My name is Mariangel Varela and I'm a MARC Fellow. In this video, I'll talk about how I happened across the MARC program, the lab that I'm currently in, the research experience I've gained so far and what I'm working on now, and I'll also talk about the work I want to do in the near future. Now I know it sounds like a lot, but that was weird. Oh well, just go with it. I'm originally from Phoenix, Arizona. I'm a first-generation college student, and my parents are both from tiny towns in Chihuahua, Mexico. They have always encouraged me to pursue a higher education. I'm an animal science major in the Ag College, and am minoring in chemistry, biochemistry, biology, and wildlife sciences. Upon realizing that I wanted to pursue research, my advisor mentioned that I might apply for the MARC program. I interviewed with Vibka Boeing, who walked me through the hallways to her laboratories, almost as if I was already part of the team. I immediately knew I wanted to learn more from her. However, I understood that our designated departments were not quite the match. But Dr. Boeing was quick to the challenge, and those differences have not stopped us yet. Dr. Boeing's lab specializes in aquatic ecology and interactions among aquatic organisms such as predator-prey or competitive interactions. Dr. Boeing has worked on every trophic level of the aquatic food web, from bacteria all the way up to semi-aquatic organisms such as amphibians and reptiles. My first field research experience was to assist a graduate student studying the relationship between the bullfrog and the Chiricahua leopard frog on a southern New Mexican ranch. Frogs are often used as an indication of the health of an ecosystem, and many scientists use them to measure the severity of anthropogenic activities. This study is a valuable contribution to the understanding of changes within predator-prey relationships due to environmental changes. For about 4 weeks, we established a routine that coincided with the activity of the frogs. I was afforded the diet analysis portion of the project. Using a method known as gastric lavage, frogs were captured, their stomach contents collected, and then released back into their ponds within the hour. When we returned, the contents of each sample was analyzed using a stereo microscope. In the samples, we discovered moths, crickets, spiders, fish, and even other frogs. This led us to conclude that not only was there a diet overlap between the preferences of both species, but there was also the possibility for predation. I was so excited with all the work we had done. I must have overwhelmingly explained to all my friends and family. And to top off my excitement, Dr. Boeing helped me with the opportunity to present these frog findings on a poster at the Chihuahuan Desert Conference. Once the ball got rolling, there was no stopping it. Dr. Boeing and I sat down to discuss what my next project would be, and after several inconclusive ideas, the final project was established and the report was made. But then the schools closed down, and all the exciting conferences, internships, and projects had to be put on hold. But never fear, for Dr. Boeing was near, and she had a project that wouldn't risk anyone's safety. I'm currently working on identifying invertebrates captured in Hesterdendi traps that were set several weeks ago at the Pitchfork Ranch. I'm also working on a separate project for a small internship at Michigan State University. This project involves combing through data sheets containing whole blood samples of various avian species, large marine life, and cats. The intent of this project is to find a range of values for mercury in the blood that is common to all individuals in that species. This way, experts may have an idea of what mercury levels in the blood may be considered normal and which are troublesome. After having experience in what it's like to work on individual projects, Dr. Boeing and I decided I am ready for a project that is geared towards my specific interests. In the fall, I hope to continue on a project that focuses on the medicinal advantages of cannabidiol. This project will focus on determining if cannabidiol reduces aggression due to isolation in mice. Cannabidiol is commonly known as CBD. It was first isolated from the marijuana plant in the 1940s and has not shown to produce any hallucinatory effects commonly associated with the plant. This, along with its healing factors, has made it a popular compound within the biomedical field. CBD has been very minimally tested for use on aggression, which plagues many individuals with PTSD, autism spectrum disorder, and more. With this project, I hope to shed some more light on this compound as it increases in popularity and use. I hope this video was both entertaining and informational. If you have any questions, I would be more than happy to answer. Thank you. Bye-bye.